Okay, so welcome to this next video. In this video what we're going to talk about is acid sensing ion channels, okay? And for short, acid sensing ion channels are often abbreviated as ASICs, ASICs, okay? So acid sensing, and there's a dash between acid and sensing, and then ion channels, okay? Or ASICs for short. So, the structure for this video then, we're going to start off with a basic discussion of the distinction of ligand-gated ion channels into three separate families, okay? So we're going to discuss the categorization of ligand-gated ion channels, because the acid-sensing ion channels are considered a ligand-gated ion channel, okay? Because they are gated by protons, and the proton is therefore considered the ligand for these acid-sensing ion channels. So we're going to discuss the categorization of all ligand-gated ion channels into three categories, so that we can then see which of these three categories the acid-sensing ion channels are going to fall into. Okay, what we will then do is a more in-depth study of the family of acid-sensing ion channels. So we'll discuss what they are gated by, which I've already alluded to, it's protons. We'll discuss the different subunits of acid-sensing ion channels, and we'll discuss what happens when they actually open. So what ions are they permeable to? Okay, right, so I should firstly give you my reference for where I took the information that I'm going to present to you in this video from, okay, because this topic isn't quite standard enough to yet be put in textbooks, so I had to go to a secondary review, a secondary research article, also known as a review, to get the information for this, so I should give you the reference for that. Okay, so the reference that I'm going to give you is Kellenberger and Grutter, which is 2014. So these two chaps, or these two people, Kellenberger and Grutter, I don't know that they were both men, these two people, in 2014, they wrote a nice review on this, okay, and that's very, very thorough. Okay, so initially when I just talked to you about the different categorizations, that's not from this review, but the later stuff on the more in detailed study of the acid-sensing ion channels is from that review. Okay, right, so let's start off then with the categorization of ligand-gated ion channels into these three giant families. Okay, so a ligand-gated ion channel is exactly what it says on the tin. It is an ion channel that is usually closed, okay, so it's not usually open. However, if you bind certain chemicals to that ion channel, it will open, and then it will be permeable to the ions, and this can change the electrical potential difference across the cell membrane by setting up currents across the cell membrane that were not present before. Okay, so ligand-gated ion channels are often abbreviated to, and I've made that mistake again now, they're often abbreviated to LGICs for short, so L for ligand, G for gated, I for ion, and then C for channel, so LGICs is often an abbreviation used for ligand-gated ion channels. Okay, so these are just ion channels which, in order to open these ion channels, you need to bind certain molecules known as ligands to them in order to cause a change in conformation, which will then allow the channel to open. Okay, right, so all ligand-gated ion channels that we find in humans can be categorized into three great families. Okay, so let's now discuss these three great families. We'll start off with family 1. Okay, and family 1 is called the P2X-like receptor family. Okay, so this is the P2X-like receptors. Okay, so these are all ligand-gated ion channels which have a similar structure to the P2X family of receptors. Okay, right, so let's see what characteristics all the receptors that are in this P2X-like family of receptors have in common. Okay, so if this is the cell membrane here, and I'll draw two lines to represent the outer leaflet of the phospholipid bilayer and the inner leaflet of the phospholipid bilayer. So the top portion This is the extracellular fluid, and the bottom portion, this is the cytoplasm. So this is supposed to represent the cell membrane. Okay, so here is our P2X-like receptor, and we're always discussing ligand-gated ion channels at the moment. Of course, that's a separate discussion, the fact that, there are many different types of receptors. Only some of them will be ligand-gated ion channels. There are also G-protein coupled receptors, nuclear receptors, and also tyrosine kinase receptors. Okay, but the ligand-gated ion channels is one family of receptors, and when we look at this family of receptors, which is the family of ligand-gated ion channels, a subfamily of the ligand-gated ion channels is this P2X-like receptor family. Okay, so they're all ligand-gated ion channels, and basically this is the pore down the middle through which the ions will move. And these ion channels are not made out of a single protein. They're made out of 3 proteins which have all been joined together. 1, 2, 3, all bunched together, joined together, and that makes the larger ion channel receptor, basically. Okay, sitting in the plasma membrane like so. So these receptors are trimers. Okay, so the entire ligand-gated ion channel is made up of 3 separate subunits which are all joined together. Okay, so what I now want to do is pull out a single one of these subunits here. Okay, which I'm now colouring in purple. So I want to pull out this subunit and have a look at its membrane-spanning topology. Okay, so I want to examine these individual subunits now, rather than looking at the entire thing, which is made up of 3 of these separate proteins bunched together. I just want to look at one of these individual proteins. So let me draw the membrane back again here. Okay, and remember, this side is the extracellular fluid, the ECF, and this side is the cytoplasmic side here. Okay, right. So the subunits of P2X-like receptors have this sort of a structure. They have their amino terminus intracellularly. Okay, then they have a membrane-spanning alpha helix. Okay, then an extracellular loop like so, and then another membrane-spanning alpha helix, before ending with their carboxylic acid tail pointing into the cytoplasm. Okay, right. So this line here represents the polypeptide. This represents the polymer of amino acid. It means amino acid after amino acid after amino acid after amino acid after amino acid, and basically it wraps round and spans the membrane twice, with the amino and the carboxy termini both within the cytoplasmic compartment. Okay, so basically all 3 of these subunits, which you trimerize together to make the entire ion channel here, they all have a membrane-spanning topology like so. So their amino and carboxy termini are in the cytoplasm, and they have these 2 membrane-spanning alpha helices with a loop in between the 2 membrane-spanning alpha helices, which faces into the extracellular fluid. Right, so to build the P2X-like receptor then, what you have to do is put 3 subunits like so together to make the full ion channel. Okay, so all P2X-like receptors will have this basic structure that they are built from 3 separate subunits which have a membrane-spanning topology like so. Okay, so there are a massive number of receptors which satisfy those criterion. The first massive family which is within this P2X-like receptor family is the family of P2X receptors itself. Okay, so there is not just one P2X receptor, it's an entire family of receptors. So there are loads and loads of different P2X receptors, and they all have this basic structure where you have 3 subunits joined together to make the full ligand-gated ion channel, and each of the subunits have the same basic membrane-spanning topology. Okay, so the P2X receptor family is a subfamily, if you like, within this family of P2X-like receptors, which is a subfamily of the ligand-gated ion channels, which is then a subfamily of the receptors. Okay, right. Importantly for us, the acid-sensing ion channel family, the A6, is also a family within this family of P2X-like receptors. So all of the A6 also have the same basic structure, where they're made up of trimers, which have individual subunits with a membrane-spanning topology like so. Okay, and we'll come back to that later. So both the P2X receptors and the family of A6 acid-sensing ion channels, they are both within this larger superfamily of P2X-like receptors, basically. Okay, right, so that's the first family of ligand-gated ion channels. Okay, and we'll come back to that obviously when we come on to discuss A6 in more detail. Now let's move on to the second family of ligand-gated ion channels. Okay, so the second family of ligand-gated ion channels is the glutamate-like receptors. Okay, and this family of ligand-gated ion channels is named after the major member, or the major members within this family, which are the glutamate receptors. Okay, so the ionotropic glutamate receptors. And when people say ionotropic receptors, that's another way of expressing the concept of a ligand-gated ion channel. So if someone tells you it's an ionotropic receptor, what that means is that it's a ligand-gated ion channel. It means that when the ligand, whatever it is, binds to this ionotropic receptor, the response of the receptor is to open a channel which will then allow ions to move through. So it's an ionotropic, having to do with ions, basically, receptor. Okay, so basically there are two major divisions of glutamate receptors. There are glutamate receptors which are indeed ligand-gated ion channels, often called ionotropic glutamate receptors, and those are the main ones that govern excitatory neurotransmitter within the brain, basically. Okay, but there are also what are known as the metabotropic glutamate receptors, which are G-protein coupled receptors. Okay, and they're not of interest to us here because we're studying ligand-gated ion channels. Okay, right, so the glutamate receptors are not the only members of this family, however. So the basic structure that you have is that you have a tetrameric structure. Okay, so if we draw the ligand-gated ion channel again, the whole structure initially, okay, like so, then we have the pore down the middle, and then the basic structure is tetrameric. Okay, so you have 4 subunits all coming together to form the entire ion channel, basically, and it sits within the cell membrane like so. Okay, so same protocol. Let's now pull one of these subunits of a ligand-gated ion channel within the glutamate-like receptor family out and have a look at its membrane-spanning topology. Okay, so I now want to pull out one of these subunits and have a look at its membrane-spanning topology. Okay, so here is the membrane. Now, basically, in the case of glutamate-like receptor family subunits, the amino terminus starts extracellularly. Okay, so here's the amino terminus. Then it has a first membrane-spanning alpha helix here, which is called the M1 membrane-spanning alpha helix. Okay, then it has what's known as a reentrant loop, which is where the polypeptide tries to span the membrane but fails and comes back out again. So this is the concept of a reentrant loop. Okay, now the reentrant loop is given a name in the subunits of a glutamate-like receptor subunit. Okay, then it's called the M2 portion. Okay, so this reentrant loop from here to here will be called M2. Then you have the next membrane-spanning alpha helix, which is called M3. So this second membrane-spanning alpha helix is called M3. Then you have the final 4th membrane-spanning alpha helix, like so, which is called M4. And then you end with the carboxylic acid terminus of the polypeptide intracellularly. Okay, so this is the structure of a subunit of a receptor which is in the glutamate-like receptor family of ligand-gated ion channels. Okay, right, now the other important thing is that the ligand binding domain of these subunits is made up of the dimerization of a portion that is within the amino terminal portion here, okay, so prior to M1, and also a portion that's within this M3, M4 loop that's between the M3 alpha helix and the M4 alpha helix. So these two portions that I've highlighted in red, those will dimerize basically, and they will then make the ligand binding domain. So the ligand binding domain is made out of the dimerization of these two red portions here. Okay, right. So that's another important feature that is conserved for the subunits of all receptors which are within this glutamate-like receptor family of ligand-gated ion channels. Okay, right. So you take 4 of these sort of subunits, stick them together, and that will make you your ligand-gated ion channel. So what we now want to see is what examples of receptors would fall into this category of ligand-gated ion channels. Okay, so firstly, the AMPA glutamate receptors. Okay, and again, the AMPA receptors is not just a single receptor, basically. It's an entire family of receptors. So there is more than one AMPA receptor. So there is a vast plethora of different AMPA receptors, and they all have this same basic structure where they're built out of 4 subunits being put together, and these subunits all have the same basic structure here. Okay, so that entire family of receptors is a subfamily within the family of glutamate-like receptor ligand-gated ion channels. In addition, you also have the NMDA receptors, which are also ionotropic glutamate receptors, so their ligand is also glutamate. Okay, and again, there is not just one NMDA receptor, there is a whole family of NMDA receptors, and that entire family is a subfamily within this family of glutamate-like receptor ligand-gated ion channels. Then finally, there is also the family of kainate receptors, which again, their ligand is glutamate, okay, and they are again a family of receptors, so there's not just one kainate receptor, there's a whole family of them, and they're a subfamily within this family of glutamate-like receptor ligand-gated ion channels. Okay, so that's family 2 of ligand-gated ion channels. Let's now move on to family 3 of ligand-gated ion channels. So, the final massive category of ligand-gated ion channels is what are known as the nicotine-like receptor family. Okay, now this by far is the most studied of the 3 families of ligand-gated ion channels, and you will see some very famous receptors within this category, okay, so you will recognize the receptors that I'm about to tell you about. Okay, at least some of them. Right, so the nicotine-like receptor family of ligand-gated ion channels has an old name, okay, which is probably more commonly seen actually, so I'll tell you about this old name. So the old name that is still in common use is to call this family the cis-loop ligand-gated ion channel family. Okay, so LGIC for ligand-gated ion channel. Okay, now some people will also just call them the cis-loop receptors, and I will explain exactly what this cis-loop is about in a moment. Okay, so basically all of these receptors that are within this nicotine-like receptor family of ligand-gated ion channels have a pentameric structure. So again, if we draw this picture where we have the ion pore moving down the middle there, basically it's made up of 5 separate subunits joined together. 1, 2, 3, 4, 5, like so. Okay, so it's a pentamer basically. So we've gone from trimer to tetramer to pentamer. Okay, right, and that's why these receptors are also called the pentameric receptors. Right, so same protocol now. Let's pull out one of these individual subunits that makes up one of these nicotine-like receptor family receptors, which are ligand-gated ion channels. Okay, and let's have a look at its membrane spanning topology. Okay, so basically, if this is the membrane again, the amino terminus of the polypeptide that makes up each of the subunits of one of these nicotine-like receptors will be on the extracellular side. Okay, then also on the extracellular side you have a loop in the polypeptide structure that is always conserved. Okay, so for all of these receptors that are going to be within this family of nicotine-like receptors, okay, they're all going to have a conserved loop here, and this loop is held together by a disulfide bond between two cysteine residues that are on the parallel strands, basically. And this, therefore, is called a cis loop. The cis means cysteine, basically, okay, and the loop refers to the fact that it is a loop. Okay, so let me just discuss the structure of a cis loop in a little bit more detail. Okay, so I'll draw the picture down here. So, let this line represent a polymer of amino acids rather. So it's amino acid after amino acid after amino acid after amino acid after amino acid, and now suddenly I'm actually going to draw the structure of one of these amino acids. Okay, and this is because this is going to be one of the cysteines which will be involved in the formation of this disulfide bridge, which is going to hold the cis loop together. Okay, so here's the amino terminal. Okay, and it will be bound to the carboxylic acid terminal of the amino acid prior to it. Then here we have the alpha carbon of the amino acid with a hydrogen coming off it, and then we have the carboxylic acid group down here. So at the moment that's the core structure of an amino acid residue. The R group then of cysteine involves a methylene group with then a thiol group attached to it. Now a thiol group is a sulfur atom then with a hydrogen atom coming off it, but I'm going to be fortuitous and not draw that hydrogen atom coming off it because we're going to have this thiol group involved in a disulfide bridge, and in the disulfide bridge the hydrogens on those sulfurs that would be on the pure amino acid will have gone, basically, so I'm not going to draw that hydrogen. Okay, now I'll go back to drawing the polypeptide as just a line, basically. So this line represents amino acid after amino acid after amino acid, and then you'll come back round and have another cysteine on the other anti-parallel strand here. Okay, so here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group. Okay, like so, and then the polypeptide chain will continue on. Now the R group of a cysteine amino acid consists of a methylene group, then with a thiol group coming off it. Now again, this thiol group will lose its proton, it will lose its hydrogen rather than its proton. It will lose the hydrogen and its electron, okay? And basically the sulfur will then bind to this other sulfur atom here, and that special bond there between the two sulfurs, that's known as a disulfide bond, okay, because it involves two sulfur atoms. So there's a disulfide bond, also sometimes called a disulfide bridge. And of all the interactions that can form between the R groups of amino acids, the disulfide links are the strongest of them. Okay, so if for instance you're doing a Western blot, you can heat your proteins up to 85 degrees to try and break the interactions between the R groups of amino acids. Okay, and it will break every other interaction apart from these disulfide bridges. So you then have to add in a reducing agent such as beta-mercaptoethanol or dithiophenol to break apart the disulfide bridges. Okay, because you can't heat it much hotter than 85 degrees. You might think, well, just heat it up higher to break the disulfide bonds, but then you'll start breaking the peptide bonds, which is something you don't want to do. You want to denature the polypeptide, but you don't want to break it up. Okay, right. So, off that side note now, that's what a cis loop basically is. It's a loop in the polypeptide structure that is held together by a disulfide bond between two cysteine residues, okay? And the three-letter amino acid code for cysteine is cis, C-Y-S. So basically you have two cysteine residues on these two anti-parallel strands, and they are forming a disulfide bond, hence why this is called a cis loop, because it's a loop that is held together by the cysteines, okay? So the importance of the cysteines warrants them having their place in this name, basically. Okay, then, after the cis loop, you then have your first membrane-spanning alpha helix, then your second membrane-spanning alpha helix, your third membrane-spanning alpha helix, then you have a large intracellular loop between the third and the fourth membrane-spanning alpha helix, and then the carboxylic acid terminus is on the extracellular face. Okay, so these membrane-spanning alpha helices are labelled M1, M2, M3, M4, so it goes through M1 to M4. Okay, and this large loop between M3 and M4 This is known as the intracellular loop. Okay, so that is the conserved structure of all the subunits of receptors which are within this family of nicotine-like receptor ligand-gated ion channels, or more commonly called the cis-loop ligand-gated ion channels. Now, why is this called the nicotine-like receptor family of ligand-gated ion channels? Well, all the other families, for instance the P2X-like receptors, the glutamate-like receptors, they were named after their most famous member, basically. Okay, so similarly, this family is named after its most famous member, and the most famous member of this family is the nicotinic acetylcholine receptors. And the nicotinic acetylcholine receptors is a massive, great family of many different receptors. Okay, so there is not just one nicotinic acetylcholine receptor, it's an entire family of receptors for acetylcholine. Now, nicotine is an agonist for some of these receptors, i.e. it will bind to them and trigger them to open just the same as acetylcholine will. Now, it's not an agonist for all of them, which you should be very grateful for if you do indeed smoke, because you have nicotinic acetylcholine receptors on your skeletal muscle, and it is these that trigger the skeletal muscle to contract. Now, if nicotine was an agonist for the type of nicotinic acetylcholine receptor that is present on the skeletal muscle, it would cause uncontrollable contraction of the skeletal muscle, i.e. it would cause convulsions, basically, and it would probably be fatal. So, nicotine is not an agonist for all of these receptors, but it is an agonist for a good few of them. Okay, so that's why these are called the nicotine-like receptor family of ligand-gated ion channels, named after these nicotinic acetylcholine receptors, which are a subfamily of nicotine-like receptor ligand-gated ion channels. Okay, right. Some other famous examples within here. The next most famous example would be the GABA A receptors. Okay, so again, there is not just one GABA A receptor, there are a huge number of different GABA A receptors. Now, glutamate is the main excitatory neurotransmitter in the brain. When it acts on postsynaptic neurons, it makes it more likely that those postsynaptic neurons are going to fire action potentials. Meanwhile, GABA, standing for gamma-aminobutyric acid, is the main inhibitory neurotransmitter within the brain. It makes it more likely that neurons will not fire an action potential. Okay, and the main type, well, there are two types of receptors for GABA, the GABA A receptors and the GABA B receptors. The GABA A receptors are all ligand-gated ion channels. Specifically, they are nicotine-like receptor family ligand-gated ion channels. So they have these pentameric structures with subunits that look like this. But the GABA B receptors are G-protein coupled receptors. Okay, right. Some other examples then of families within the nicotine-like receptor family of ligand-gated ion channels is you also have the 5-HT3 receptor family. Okay, and again, there is not just one 5-HT3 receptor, there are multiple 5-HT3 receptors. Okay, so it's a family of receptors. Now, most receptors for serotonin, which is also called 5-hydroxytryptamine, or 5-HT, are G-protein coupled receptors. Okay, but the 5-HT3 family of receptors are ligand-gated ion channels, and again, these are ligand-gated ion channels that are within this family of nicotine-like receptors ligand-gated ion channels. Finally, there is also the glycine receptors. Okay, so within the spinal cord, glycine is an important inhibitory neurotransmitter, and the receptors for that are ligand-gated ion channels, and there is an entire family of receptors for glycine, okay, and they're called the glycine receptors, and they are all nicotine-like receptor family ligand-gated ion channels. Okay, right, so that's the categorization of ligand-gated ion channels into these three superfamilies, basically, okay, based on their core structure, i.e. how many subunits bind together to make the whole ligand-gated ion channel, and what the membrane-spanning topology of those subunits are. Okay, we have now seen that the acid-sensing ion channels, the A6, are within this P2X-like receptor family of ligand-gated ion channels. In the next video, what we'll do is discuss the acid-sensing ion channels in more detail.